I'm delighted to introduce the moderator of our second panel, Dr. Maha Farhat. Dr. Farhat is the Gilbert Omen Associate Professor of Biomedical Informatics at Harvard Medical School, and she's also an associate physician in the Pulmonary and Critical Care Division at Massachusetts General Hospital. Dr. Farhat is a pioneer in developing creative computational approaches to associate genotype and phenotype for infectious disease pathogens. She's also a wonderful colleague of mine at DBMI, and she's going to lead a discussion on bringing genomics into public health.
Thank you so much, Dr. Manrai, for the kind introduction. It's really my sincere pleasure to moderate this session. The second panel, where we're going to be discussing how we can bring genomics into public health practice. And this is a topic that's very near and dear to my heart. As Dr. Manrai mentioned, my group is very interested in the application of short and long-read sequencing technologies to infectious pathogens, and specifically to look at pathogen evolution, antibiotic resistance phenotypes, and transmission inference. And so, really, I've seen in the last 10 to 15 years that I've been in this space, we've really seen a massive uptake of the use of sequencing technologies in microbiology and population genetics research. Mostly it has been focused on, but then coupled with the really small size of the pathogen genomes, it's really been possible to scale up the sequencing very early on, from thousands to hundreds of thousands, to most recently for COVID, we have over 16 million COVID genomes available in the public space. And the sequencing of the COVID genome in particular is a really good example of how sequencing can better and improve public health, because it's the release of that first genome very early on in 2020 that enabled very rapid vaccine development at the time, and really enabled some early surveillance efforts. And then probably even more so for maybe human genetics, where we're still in the realm of research and trying to apply genetics for specific diseases. For pathogens, sequencing lends itself naturally to public health applications because it allows us to study very important public health threats like antibiotic resistance, like understanding epidemics and disease spread patient to patient, and using that information to try to control disease. And so these kinds of innovations have enabled and currently enable tracking, for example, of the flu genome season to season, predicting or using information on the last season's viral genomes to design the next vaccine, and continue to survey the accuracy of our diagnostics and track antiviral and antibiotic resistance. So in the next hour, we will focus on the current state of using sequencing for surveillance of existing and emerging infectious disease threats. We're also going to be we have a representative from industry who's going to be tackling the topic of using innovative molecular tools that could be deployed in potentially resource-limited settings to accelerate other types of human disease surveillance. And we will focus on the past, present, and future of these technologies, its success and challenges, and also access and equity. So it's my pleasure to introduce our first panelist, Dr. Garrett Frampton, who's the Vice President of Computational Discovery at Foundation Medicine. Dr.
Hi, everyone. I'm really grateful for the opportunity to talk here today. I can give a quick introduction of, I guess, Foundation Medicine and myself. Foundation Medicine is a company that was founded in Cambridge, Mass, 2011. And the goal of the company was to translate a lot of the advances that have been done in academic research, particularly stuff like the Cancer Genome Atlas, other research projects, to bring that technology and those discoveries to patients with cancer, first across the United States and now across the globe. I was trained in chemistry and biology and did a graduate degree in biology, all computational biology. I worked a lot on building the first versions of our assays, and the last, say 8 years or so, I've led a team we called Computational Discovery, doing research on the data that we collect. Just a real brief idea of what we're doing. I think most of the sequencing that we're talking about here is, other panelists are talking about germline sequencing or hereditary genetic markers. At Foundation Medicine, we take DNA out of cancer specimens or take DNA out of blood from patients with cancer and sequence a small set of a few hundred genes that are known to be relevant. And we're mostly looking for somatic mutations. So these are mutations that have arisen during the course of a patient's progression of cancer. We do see hereditary causative mutations as well, but that's just one distinction I wanted to make. Yeah, the team I lead, computational biologists looking at the data. We're trying to do sort of short-term product improvements, so seeing the test is working okay, but figuring out ways to get it to work better, more accurately, more robustly. Long-term product improvements, so bringing new biomarkers that perhaps we didn't realize we could measure when the tests were created, and so new information for cancer patients. And then basic research. I feel really fortunate that I can't exactly explain why, but in an industry setting, we get a great opportunity to publish the findings that we're making to advance just the general state of knowledge in the field. So that's sort of the three sorts of things that we're working on. We're working mostly with DNA sequence data, but we're also working with pathology images. Sort of big data sets is what we get excited about. And in keeping with the theme of the meeting and borders, I just made a list of all the challenges related to borders that I've come across in the sort of the goal of bringing high-tech DNA sequencing technology to cancer patients around the globe. Identified data like test results, clearly with patient names on it, identifiers, that can be challenging to move across borders. De-identified data for research purposes, the rules about what counts as de-identified and what you can do with it vary across borders. Genome sequence data itself, it's not the test results, can't really be de-identified. So how do you do research with genome sequence data across borders? Software, so we build software that processes the data and gives meaningful test results. There's challenges about deploying software across borders. One of the interesting things about our assay is that the analysis is actually kind of a combined human and computer. We let the software do 99% of the work, but the tough 1%, we have people as actually part of the medical device making decisions. How do you translate that across borders? That's a real challenge. Biospecimens, so clearly shipping blood or tissue biopsies, tumor biopsies across borders is a challenge, and sending it back so that additional tests could be run. There's laboratory equipment and trained personnel. You can either ship the specimen or ship the lab. Those are both solutions we've tried to do to varying success. So the laboratory and the trained personnel, you need those to be able to translate across borders. The reagents and kits are not universally available, and so figuring out or maybe you might say, well, rather than building a lab, can we build a kit and ship a kit? That's another solution to crossing borders. So that's an interesting thing. There's medical diagnostics have regulatory approvals, and regulatory approvals are done by different agencies in different parts of the world. So you might think, well, you built something great, works really well, but you actually need to have that approved in lots of different geographies. Similar but separate is evidence that the test works, right? Evidence of diagnostic accuracy, analytic validation, sort of the technical term. That evidence is not perfectly transportable as well, and the desire for different types of evidence in different places is very natural and normal, but something to contend with. And then the evidence of benefit to patients. I don't think we should assume that all biomarkers are perfectly transportable across borders due to differences in patient populations. But even if they are, the accepting of that evidence as being worth demonstrating benefit to patients might be interpreted differently in different geographies. So, and that's just the, let's say, technical challenges. There's also huge challenges with resources to do the diagnostics test. And unfortunately, I mean, it's fortunate that we have great new drugs, but great new drugs tend to be expensive. So if you have a diagnostic test that recommends a drug that's not available to a patient population, that's not really going to actually benefit the patient. So a lot of, when I left graduate school 13 years ago, went to Foundation Medicine, I had no idea all the stuff that I'd be getting into in terms of technical and these sorts of implementation challenges, but it's been fascinating and not at all tired of it yet. So thanks so much for the opportunity.
Thank you, Garrett. I'd love to introduce Dr. Jennifer Gardy, who I know from the world of TV, and she's been wearing many hats, but she's currently Deputy Director of Malaria at the Bill and Melinda Gates Foundation. Welcome, Dr. Gardy.
Thanks, Maha. It's a pleasure to be here. My background, Maha said, we've worked on TB together for a long time before I joined the foundation. It's really around pathogen genomic epidemiology and how do we use DNA sequencing as a tool to better understand issues in public health, in epidemiology. But I've been with the foundation now for 5 years, and my portfolio within the malaria team really boils down to, at the end of the day, ensuring that national malaria control programs and the other stakeholders, whether it's WHO, whether it's the Global Fund, whether it's us as the foundation, do we have access to high quality data with which we can make good programmatic and policy decisions? And it includes a lot of work, everything from strengthening routine surveillance to building out quantitative data science capacity within and adjacent to malaria control programs. But a good chunk of the work is on using genomic technologies to answer questions in the malaria space. So we call it our malaria molecular surveillance portfolio. And it's been really exciting to see it grow over the 5 years that I've been with the foundation. When I joined at the beginning of 2019, we had 2 investments totaling about $5 million in malaria genomics, and both of those were to institutions in the global north. But over the last 5 years, we have changed that dramatically. Our primary goal has been moving the work to malaria endemic countries under the leadership of African scientists, and we've seen that portfolio now grow to nearly 50 grants totaling about $100 million. Over half of that amount in total goes to malaria endemic countries. If you look just over the last 2 years, 2/3 of our funding has gone to endemic country scientists, and almost half of the PIs or co-PIs leading those initiatives are African women scientists too, which we're really proud of. So with my couple of minutes here at the start of the panel, I thought I'd just call out 3 tenets that have guided how we have shaped that portfolio, how the strategy has developed, what sorts of investments that we've made that I think have made a real difference in allowing us to see that degree of impact. The first is that all of the work that we've funded in this space has to be done in service of epidemiological, actionable decision-making. There's a lot of stuff that you can do with genomics. You can understand the evolutionary history of organisms, you can look at population structures, et cetera. But we are funding genomics work that is going to lead to changes in programmatic guidelines, in policy. There are other donors out there that aren't the foundation that are very well positioned to fund some of that basic science work, but we want our dollars to go to molecular surveillance. And when a national malaria control program sees impact, they see the value that genomics is bringing to their program, that really helps to create the appetite in other country programs to be doing the same. So by investing in results that drive, decisions about updates to treatment guidelines or decisions on, switching to a new rapid diagnostic test, when you have country programs that have demonstrated success in that space, they will evangelize to other country programs, and you can help to build this momentum. It becomes sort of a positive feedback cycle. The second point that I wanted to make, which I think is related to that first one, is that the work that we fund, many times it is being done by scientists that are working in either an academic institution or a very sort of translationally oriented research institute. And those people really need to be working from day zero of a project with the National Malaria Control Program or the other person that is the end user of that information, the people that are going to turn the insights from the genomic studies into a decision about commodity choice or an update to some sort of guidelines. So what you're starting to see, across sub-Saharan Africa, there's a lot of genomics capacity starting to be established within national public health institutes, within ministries of health. COVID really accelerated this, and Africa CDC has played a huge role. But still, the lion's share of the work does still sit in those academic centers. So we really work hard to ensure that our genomics experts are connected, as I said, right from day zero with the folks in the National Malaria Control Programs that will help them co-create questions, that they'll come to those researchers and say, look, the thing that we really need help with here is a national survey of the prevalence of HRP23 gene deletions and what that means for our choice of rapid diagnostic tests. So co-creation is key. And the third thing that I'll end on is I think it's been really important for us to consider that we're not just investing in individual projects, but we need to be investing in an entire enabling environment, an entire ecosystem. So you can't just put a sequencer into a lab and expect immediate public health impact and for that impact to be sustained. There's a lot of stuff that you have to be investing in addition to those projects. So you need to be investing in people. Obviously, this is training the next generation of scientists, but it's also giving leadership and professional development training to early and to mid-career investigators and helping amplify their voices on global stages and get them in more leadership positions. It's investing in common platforms. It's investing in ways for groups to share SOPs with each other, to share bioinformatics workflows, even things like building out local capacity in the bioengineering space so that you've got trained technicians who can come in and service some of these equipment. You're looking at things like investing in local manufacturing capacity. So are there groups in malaria endemic countries where they are capable of producing PCR reagents in-house? Supply chain and procurement issues are always a challenge when you're working in lower middle income countries, so really working with agencies like Africa CDC to support things like pooled procurement and delivery of reagents. And it also means working with groups like WHO, for instance, to ensure that there is normative guidance out there that countries will follow that says, hey, we strongly recommend the use of a genomics approach for use case X, Y, or Z. And it also involves working with groups like the Global Fund, who Crystal had mentioned in her opening keynote. They are the major funders of malaria and then HIV and TB responses in the Global South. And so we need to be demonstrating to the Global Fund that supporting labs and lab strengthening is a vital activity too. So there's a lot of things that you have to support in that enabling ecosystem beyond just funding really cool science. There's about a million other things I could talk about, regulatory affairs, the need for ethics and legal and social implications of this work and scholars in that space, creating community, all sorts of stuff like that. But I think those three that I just gave you would be my top lessons learned over the last 5 years. Thanks.
Okay. Thank you so much, Jen. It's my pleasure to introduce Dr. Mark Lipsitch. He's a professor of epidemiology at Harvard Chan School of Public Health and also the director of the Center for Communicable Disease Dynamics, in addition to additional hats at the Centers for Disease Control. Thank you, Dr. Lipsitch, for being here.
Thanks, Maha, and thanks to the organizers for the invitation and to all of you for being here. A little bit of background about me. I have led the Center for Communicable Disease Dynamics at the Chan School since 2009, and a couple of years ago had the chance to help start up the new Center for Forecasting and Outbreak Analytics at the CDC, which is the newest center at the CDC and is focused on forecasting and analytics, as the name suggests, not to replace all of the program-specific work that's going on at CDC, but to provide a surge capacity and a really hardened industrial strength capacity for those kinds of analytical work during health emergencies, including pandemics, but not only pandemics. And I should say that I'm speaking in my Harvard capacity, not my CDC capacity today. I'm going to talk in my comments and as we discuss later, mostly about the COVID-19 experience and possibly make some references to one of my earlier loves, which was antimicrobial resistant bacteria. I think with COVID-19, we've really seen that sequencing is an important tool within many different aspects of infectious disease surveillance. And I really liked what Dr. Gardy said about the sort of very important, the high importance of making sure that data are used in public health for applications and for decision support and not just for curiosity. Curiosity has an important place, and it's certainly where I come from as a scientist. But I think that really being thoughtful about the design of systems for surveillance and how to incorporate genomics into those systems is critical to making the most of them and having the data that we need in the form that we need it in order to support those decisions. And so I'm going to draw on some of the experiences in the first couple of years at CDC in some of these remarks. So I think that surveillance for decision support of the sort that Jen just described could sort of be broken down into 4 or 5 activities: detecting new things, characterizing them, tracking them, and estimating the burden in space and time, and controlling them. And the 5th one, which is perhaps more on the science side, is studying them and trying to understand historically what has happened. But in the more sort of present and forward-looking pieces, each of these has particular requirements, and the role of sequencing data in each is really distinct. And I think often when we think about surveillance, we think of one thing. We say we have a surveillance system. But in fact, at least for pandemics and equally for resistant bacterial infections, you really need different systems purpose-built for these different roles. So in the detection area, we really think about casting a wide net. We're not interested in representativeness. We really would love to see airports and travelers and the people who may be introducing something from one place to another, and we don't need to know every person in the population. We need to focus efforts on places where we're likely to discover things because we're interested in, is it there or not yet? And so we want to bias those systems really just deliberately towards where we're likely to find something, but try to be creative in thinking what that is so that we don't focus on so we don't just prove our own prejudices. Technologically, of course, that means that metagenomics and other approaches that are less detailed but much more likely to pick something up are important, with the caveat of preventing false positives. I think some of you will remember the New York Times' breathless coverage of the anthrax in the New York City subway a few years ago, which turned out not to be anthrax. So false positives are a concern with these detection efforts. In terms of characterizing, the goal is to really understand the properties of the pathogen, whether it's how severe the infection is, and whether it's susceptible to various countermeasures such as vaccines, monoclonal antibodies, drugs, and so forth. And in this case, I think the crucial piece is really to connect sequence data to other data, and that is a lot easier said than done. If there's one critique I would make of our existing US infrastructure for sequence data right now, it's that the number of sequences is enormous, but the number of sequences that are collected in a systematic way from which you can make clinical or diagnostic or other kinds of inferences is much smaller. And I think malaria programs provide a good example of some of the way forward, and I think that field has been thinking about this for longer, certainly than COVID-19, which didn't exist on our minds, at least until very recently. In terms of tracking, the goal is to really understand the burden of the infection and which strains are causing trouble. And the really important things are to have a good sampling frame so that you're tracking a defined sample of the population that may be in some way representative of that population. The UK has been a real leader in doing that, in connecting some of their sequencing efforts to very systematic surveillance systems that are representative of the population, such as the REACT and COVID infection surveys. In terms of control, the questions are kind of knowing how to treat a new infection and prevent its spread. And there, the role of sequencing is more particular. It's not always necessary. Sometimes you just need to know that it's there, not know every detail of it. But I think the AMR example now is a nice place to think about that. So in the case of antibiotic resistance, indeed one of the most important goals of sequence-based surveillance is knowing how to treat. And in that case, one of the interesting issues that occurs actually in all of our areas, but with few other diagnostics, is the idea that the target is moving. If you invent an assay for blood glucose, you can use that assay today, and you should be able to use that assay 100 years from now, and it will give you the same answer with equal validity, roughly speaking. But if you're using a sequence-based diagnostic to try to figure out whether a pathogen is resistant to a particular drug, 100 years from now, these pathogens will have invented all sorts of new ways to be resistant to the drug, and they will have had compensatory mutations that may change the meaning of the sequences we know about now. And then last, I think there are lots of perhaps a little bit more retrospective studies that can nonetheless help us to design our future work. So, for example, sequence data is very helpful for phylogeography. When you assemble a tree, an evolutionary tree of the sequences you have, and lay onto that some data about the location of those sequences and of their putative ancestors in the tree, you can make inferences about how different pathogens move across the world or across a particular country, and, for example, test the hypothesis, as we did a few years ago with Jonathan Grad, that particular resistant strains of bacteria have moved from west to east in the United States. And it's not just that we detected them first there, but they were actually there first and then later spread. Jonathan Grad is a valued colleague here at the Harvard Chan School, and he and I wrote a paper back in 2015 in which we said, I thought as kind of a throwaway comment, that the epidemiologic line list, the list of demography and clinical outcomes and other things that we collect about as epidemiologists about disease cases, should have a new column, a very wide column, which would be the pathogen genome sequence. And I thought it was a throwaway because it seemed such an obvious thing that connecting data to epidemiologic data to genomes would be a valuable thing to do. It still remains the exception and not the rule. And that's a bit of a disappointment to me. So I think we have a lot of work to do to maximize the value in public health of these genome sequences by attaching them to data and by understanding why we're sampling and then sampling appropriately for purpose. And I think it's great to have this panel because I think some of the other, particularly the malaria field, has thought about a lot of these problems already.
Thank you very much to the panel for this wonderful introduction. And I just want to remind everyone on Zoom, you're welcome to post questions as we go along. Please go ahead and do that. And if someone can alert me to the presence of these questions, please go ahead. I would like this to be a dynamic session where I'm not the only one doing the asking. So I'd love to hear from you. And in addition to the audience in the room, certainly feel free to come up to the mic. But I will go ahead and get started asking some questions around this. And it was really a wonderful summary that we heard from Dr. Lipsitch around the goals of surveillance, that we have many forms and flavors of surveillance, including detection, characterization, tracking, control. So I wanted to start the panel off by asking, how would you contrast sequencing with existing or more traditional methods of doing these surveillance? And how would you say the major innovation of sequencing has been in terms of improving upon past technologies?
I can take a start from the malaria perspective. So I think where we are now in malaria, if you're looking at malaria endemic countries where there's still quite a high burden of disease, their routine surveillance systems are still growing. I mean, I worked for many years before joining the foundation at British Columbia's Center for Disease Control, and our surveillance systems were far from perfect there. But looking in endemic countries, many of them only recently started building out malaria-specific data repositories or integrated malaria information systems and having those systems feed into national level health management information systems like DHIS2. So as those routine surveillance systems that are taking in data from, community healthcare workers, diagnosing people with rapid diagnostic tests, we're taking in data from hospital labs, doing microscopy from other healthcare facilities, and integrating all of this up. As those systems are still growing and becoming stronger, molecular data provides a really useful complement to those. So to give you one example, one of the things that we typically do in order to estimate malaria prevalence is a survey in school children. It'll be sort of a nationally representative survey where kids under 5 or sometimes kids under 10 get a little finger stick, and we simply look for the presence of parasitemia. And those surveys are incredibly resource intensive to do. So countries typically might do them on the order of every 3-4 years. And so when you're looking at prevalence estimates for a given setting, you could be looking at data that at that point is 3 or 4 years old. But if you start to look at things like parasite genetic diversity, it actually turns out that that diversity, whether it's at the population level or whether it's multiplicity of infection within a single host, can actually give you a pretty good surrogate measure of transmission intensity, which you can then extrapolate into measures of prevalence and incidence. So it gives you, at the moment, kind of an alternative source of epidemiological intelligence that can really complement your routine surveillance activities and make up for any gaps in that. But then there's also spaces in malaria where molecular surveillance is the only way that you would be able to answer a question. So if you're looking at, for example, I mentioned earlier HRP23 gene deletions. If you have a parasite carrying these deletions, people will test negative on most of the current rapid diagnostic tests, even though they actually do clinically have malaria. So the only way to actually estimate the prevalence of these deletions in your population is with molecular methods. And if your deletions, if you're over a 5% 5% threshold, that is the trigger to change to a different type of RDT. So there's some cases where molecular surveillance is absolutely necessary, and you would never get that information otherwise.
Thank you.
I'll just add, I think in other contexts, very similar things occur. I think it's a realistic approach to think that surveillance data are a series of approximations. The systems are never perfect. And one of the nice things about genomes, as Jen said, is that they give you a kind of orthogonal picture of what's going on. And the field of population genetics that sort of flourished starting in the early 1900s had a lot of really clever people doing very academic, seemingly useless things, but many of them with quite specific practical goals with animal breeding and the like, but also a lot of very complicated calculations and clever ideas about what you can learn about the history of a population and the current state of a population based on the genetic state of even a few loci. And so that has paid dividends in the current time because now we can look at a few loci without much trouble, even if we don't have whole genomes, and we can learn even more when we do have whole genomes. So in a way, we're kind of like reaping the benefits of all that clever work that happened starting about 100 years ago. An example maybe is the use in both the 2009 flu pandemic and in COVID of coalescent methods where people use a small number of sequences from, and those were developed later, not in the early 20th century, but more recently, those methods allow you to take a very small, relatively small number of genomes and reconstruct how fast the population must have been growing based on the structure of the evolutionary tree that links them. And that's a nice example where you may not have surveillance out there that's really capturing every case, and you can't be sure that if you're counting how many cases we detect on day 1, day 2, day 3, that's not all about growing surveillance capacity rather than about actual changes in the numbers of cases. It's probably both, and you don't know the mix. But with genomes, they don't lie in that respect, and there are a lot of things you can learn as a sort of second source of truth. And then the last thing to say is that we've also been very lucky with COVID-19, bizarrely lucky that as the different variants of COVID-19 have swept around the world, we've had the chance, we've had the quirk that they have been alternating in their phenotype on a particular PCR test, the so-called S-gene target failure. So it's been possible to estimate which variant you're dealing with without a full genome sequence at each of the, almost all of the major transitions between variants. So we've had some good luck there, which has made it possible to do at lower cost and more broadly something that we would otherwise require longer sequences for.
Thank you.
So in the cancer space, it's also a satisfying answer, pretty different. The materials that are available for doing diagnostics are very small, like a needle biopsy of a lung tumor is, minuscule. And so the massively parallel nature of sequencing lets you assess all the relevant biomarkers from just one specimen. And I guess you don't know everything about a cancer by sequencing its genome and its epigenome, but you know a real lot about the molecular state of that cancer. And so from just one very small specimen, you can get an enormous amount of information, which if you could get that much information in proteomics from one specimen, that would be great, but the technology isn't available to do that sort of degree of parallel assays.
So would you say then in a resource-limited setting, would that address an unmet need for, say, cancer screening or cancer surveillance?
I mean, if that massively parallel technology can be done economically, yeah, and I think, as we've seen, the price of sequencing is going to zero. I mean, it's not getting there fast enough, but I'm optimistic that that won't be the limiting factor.
I don't think you're the only one who's optimistic. I think we have a question from the virtual audience.
Yes, this question comes from Amy Altan from the Department of Medical Informatics here at the school. Okay, I think we're good. Okay, yes, from Mimi Alcatan from HMS DBMI. Mimi says, my perception is that there has been a lot of progress made in molecular surveillance in a number of different fields, including public health, medicine, civil engineering, and epidemiology. Is there a way to get these fields to better work closely together as they often become siloed?
I would say that they are beginning to work well together. I mean, I think the advent of wastewater sequencing and quantification in public health is certainly one example. I think maybe an example where they haven't been as integrated is the one that I gave earlier, where the sequencing and the clinical and and demographic data often aren't linked. And so there are kind of two approaches to solving that problem. One is the high-tech approach, which is privacy-preserving record linkage and various informatic efforts to take two disparate data points that actually pertain to the same event and link them. And the other is really just bringing sequencing to the point of care, or at least to the immediate diagnostic environment, so that the data get linked directly. But I don't perceive it as that siloed, but maybe others do.
I'd say lightly siloed, getting better. I think one of the examples where there's room for improvement, earlier I mentioned the Global Fund, which supports responses to HIV, TB, and malaria. And it's not unusual to go into an institution anywhere in this world. This is by no means exclusive to, say, sub-Saharan Africa. But you go into an institution and you find one lab that has a sequencer that's running for TB, and you have another lab down the hall who's running a sequencer for HIV, and the two PIs rarely connect with each other. And it would be great to move to more of a core facility model where you have embedded in a national public health institution a group that is just sequencing whatever happens to be coming through the door that day. I think we're going to be getting there with things like Africa CDC's pathogen genomics initiatives, and we will get to a place where programs around the world focused on genomic epidemiology might look a little more like they do at CDC, like they do at Public Health England, where one core laboratory is handling the analysis for a range of pathogens. I think the other opportunity is really going beyond public health and pulling in experts from other domains. Like I see in the bioinformatics space, bioinformaticians kind of writing their own code, and I can say this as a bioinformatician, we're not good at coding. If we want to have a platform that is accessible to users around the world, particularly users who aren't trained in bioinformatics, you need something that's being really well designed by a group of software engineers. So engaging the software development field in bioinformatics would be huge. Engaging organizations that work on logistics, getting building materials, getting military troops out to places would be great to engage with on supply chain issues. So really thinking beyond the public health box and engaging other sectors, I think is a new and exciting frontier.
Yeah, just a quick piggyback on the bioinformatics question. Publications that share methods, share software, share information help. I don't know if it's exactly breaking down silos, but it's distributing information in a way that, a lot of the new ideas we have for algorithms don't necessarily come from the cancer space. So if we didn't see those methods, we wouldn't have the opportunity. So it's a lot of work to share software that's good and helpful, but don't be afraid. You don't have to be perfect.
Yeah, the the discussion triggered one other thought, which is I think one siloing issue that I've observed is a kind of two cultures issue between laboratory folks who in some public health agencies are the locus of sequencing work and epidemiologists. I think, in the spirit of trying to add the sequence to the epidemiologic line list, we are doing the same thing. I mean, we're doing it with different types of data, but the lack of understanding, which I think is due to the way we train people, that they are either genome jocks or they're epidemiologists, but not both, is a problem, and that's something we work really hard on in our Center for Communicable Disease Dynamics, the group here at Harvard, to try to cross-train people so that they're not scared of whichever one they're interested in. They're not scared of the other one, and they see it as an extra tool rather than as like some kind of foreign data.
Please go ahead.
Thank you. Hi, I'm Esther Abels, and I'm really enjoying this intriguing discussion. It's very insightful. And I have been working in the past in pharma, and I've always been interested in what genomics, how they could change medicine, actually. But recently, 10 years ago, I started to also work in histopathology, so more the phenotyping role. And as you mentioned, the target is shifting, the target is moving. So I was wondering, to build on the data linkages, if you could have access to also more of that data with the phenotyping, and then linking it more to the genotyping, as well as to all the demographics, etc. What would you think that could bring to medicine? That's one. And two, also, could you elaborate more, like, where should we start with what data is most important to link? Is it the demographics? Is it the phenotyping or anything else, radiology, for example? Looking forward to hear more about that.
I can say in the cancer space, it's knowing what happened to the patients after they interfaced with us as a diagnostic test or, a therapy selection. There's not, you can make a guess about molecular target and therapy and see that a trial 5 years ago showed that should work, but being able to confirm with patient outcomes, that's the least, the most challenging, I think, type of data for people like myself to access and use.
In the malaria world, one of the good examples is the use of molecular analysis in therapeutic efficacy studies. So these are essentially the cornerstone of countries being able to understand whether their current drug regimens are working or not. And so essentially they enroll folks with clinically diagnosed malaria at different health facilities. They will put them on treatment. They'll follow them for an extended period and do blood samples at multiple time points throughout that, and they'll look for cases of treatment failure. Where was this person, taking their drugs? Were they adherent to the treatment, which is only a 3-day course of treatment? Did they take the treatment, and are they still showing up with parasitemia? So the molecular work is really important, first of all, to distinguish, is the reason that they're still showing up with parasites at day 42 because the original treatment failed and their original infection still persists, or is it a new infection because they live in a really high transmission area and just happen to be reinfected? But then if it is treatment failure, you want to do molecular follow-up on all of those and figure out which resistance marker is driving that. So obviously you need to know what medication regime these folks were on, but it also helps to have information about space and time, because we're really interested not only in these individual treatment failures and what that means for treatment guidelines, but what does this mean for the regional epidemiology of drug resistance? So we're starting to see resistance to artemisinin emerge in multiple foci in East Africa now, and being able to track the spread and the clonal expansion of some of those populations is becoming really, really important for us. But because it's still very much a population level surveillance tool, we can get away with surprisingly minimal metadata and still derive some really useful conclusions.
Thank you for your informative discussions. So on the problem of applying genomics technology into public health and population science applications, so my question is that what might be the future endeavors? Microscopically, for example, what might be the single molecule sequence technology like nanopore and single cell sequencing technology and paper-based diagnostic technology can contribute to these endeavors, and microscopically can public health surveillance systems be integrated with other environment protection systems framework and public security framework.
Quite a few questions in there. There was single cell, yeah, no.
Yeah, I mean, I'm very excited about being able to get that information out of the talking about single cell and single molecule sequencing. I don't know that the technology is quite there yet. So I would love to be able to get that sort of data. More information equals better predictions and more precision, but somewhat technology limited. I didn't totally understand the second part of your question.
The public health surveillance system, like genomic sequencing platform, can it be integrated into environment protection architecture in the nation or public security architectures?
I think maybe referring to biothreats, or is that what your environmental risk and biothreats and pathogen or other disease?
Pathogens.
I don't know much what to say to that question, but on the first one, I think one thing about nanopore sequencing that's been very important in a lot of my colleagues' work has been the ability to answer questions at speed. So in principle, you can start to get reads from within minutes from a nanopore, and it won't give you everything. But some colleagues at DBMI and Harvard School of Public Health developed an approach to looking for drug resistance in several bacterial pathogens, not by finding the resistance markers where you have to actually sequence the right part, but by similarity, by evolutionary similarity of the sequence you do detect, whatever it happens to go in first, to known resistant clones. And so I think that's an example where speed is really important, and the single molecule approaches have that advantage. I think also just the portability and my understanding is that much of the Zika outbreaks over the several years that they were prominent were studied through nanopore sequencing just because of the portability and ability to get things to where you need to be.
Yeah, I think
I guess to the biosecurity question, a quick answer might be that I think when it comes to sort of engineered bio-threats and genomic surveillance for those, well, it's not an impossible possibility. It's a rare possibility. And really, what we need to be looking out for when it comes to biosecurity is just the threats that Mother Nature and evolution will bring to our doorstep on a fairly regular basis. So I think when you can look at things like nanopore sequencing technology, which are highly portable, affordable, and easy to bring into the field, it gives you a unique opportunity for doing routine surveillance, likely with metagenomics, as Mark mentioned earlier, at those interfaces where you are likely to see spillover events that then become potential epidemics or pandemics. So I've always thought that looking at humans who work at the human-animal interface and regularly setting up some sort of sentinel surveillance system in those populations is probably going to be our best bang for our buck in looking for emerging threats.
Please go ahead.
Chesley here. I had a question for you, Garrett. Just thinking through as we move into a phase
I'm
where...
not sure your mic's on.
Yeah,
The people
sorry about
online
that.
probably
I wanted
can't
to,
hear.
when we move from a phase where we're doing testing now for diagnostic purposes, and now later on as we begin to do that longitudinally, so, adjunct therapy, after therapy, and then we begin to go back and sequence a second and third and fourth time, and then beginning to look at those markers across from a profile perspective. I'm just curious how Foundation is thinking about doing that, and what is that exciting kind of work for you, and is that something that your team is working on?
Yeah, so right now, pretty much each specimen and each diagnostic test result is one specimen at a time. I think liquid biopsy, DNA out of blood plasma, is going to enable patients to get multiple genomic diagnostic time points. Right now, that's reserved for people with pretty large resource pools. I think there's probably a little bit of a paradigm shift in how do you, right, a diagnostic test result is signed by an MD on a particular specimen. We probably need a space for diagnostic test results which are multi-time point or comparison time points where the actual diagnostic result needs both time points and needs to be signed off on by both time points, which means the information needs to be preserved in a way we're sort of not set up for. So there's a real big logistic and sort of medical workflow and data, let's say security or data provenance challenge there. So I'm excited for it. I think there's a lot of potential there, but how are we thinking about it? I think we're worried more right now about getting a single genomic time point to as many cancer patients who might benefit from it as possible. But yeah, it's definitely coming. And I think like a lot of stuff in this space, it will be maybe not the most cost-effective way of approaching things at first, but it'll get there. It's my prediction.
Thank you.
I think I'll take the last question. All right. Sorry to those who had their question online. So I just want to close this off because we're running out of time. But I think there's a really important, hopefully it's not going to be quick, but if we can have an abbreviated discussion. I was hoping we could discuss how to ensure that genomics doesn't inadvertently create or exacerbate public health inequities, especially in regions with limited resources or marginalized communities. I know this is a loaded question, and there's only 3 minutes, so.
I don't know, I can start by saying I think what is central to remember is we really need to make sure that communities and countries are in the driver's seat for this sort of work. In Crystal's presentation, she talked a bit about some of the risk communication and community engagement work that Target Malaria has done. And honestly, I mean, if she were just to focus on that, she could fill a day with the incredible work that's gone into, from day zero of the project, working with communities and potentially affected populations to co-design an equitable future for this gene drive technology. So I think putting communities in the driver's seat, we've seen a nice example of this across sub-Saharan Africa. There's a group called the Pathogens Genomic Diversity Network, and one of the things that they do is bring National Malaria Control Program managers who may not know much about genomics, but they bring them for training in a lab in Mali, and over the course of a couple of weeks really introduce them to the technology, what it can do, and it allows them to then go back and say, this is something that I want to incorporate into my program. So yeah, co-creation and putting communities and programs first.
Thank you.
Pretty high level, but if if you have disparities with haves and have-nots, if the haves are willing to invest money into helping not just the haves but the have-nots, that would go a long way. There's a lot of ways to do that, right? There's providing therapies at less cost. There's including patients in clinical trials that might not be the cheapest way to get the trial done, but it's going to be more beneficial to more people. So,
how do you incentivize that?
I don't have a great answer for that, but shame? I don't know.
It's a challenge. I don't know, Mark, if you want to comment.
Hard to top those responses, but I guess I would just say that with acute pathogens in, say, the developed world, it should be clear to everyone that the threat applies to all of us, and it doesn't apply equally because of existing inequities. But if you look, for example, at the spread of COVID-19 in Chile, it started with people who had been on ski trips. And so you look in the richest neighborhoods of Santiago, it's there first, and then it spreads and does its damage in the usual socioeconomically reinforcing ways that it does. But it comes there first. So it is the case, whether we like it or not, that we all benefit from knowing what's in the community and looking all through the community to find it and prevent its spread. How to fix people's perceptions and help them appreciate that is a problem beyond our scope, I would say.
Thank you. I just want to say that, I mean, in the spirit of improving public health and the fact that genomics can and is a tool in our toolbox, it's not the only tool, it's one of the tools, and if it's going to help push forward the agenda of more access to care, better surveillance for all, then that would potentially help us improve equity. So thank you all very much for a wonderful panel, and thank you all to the audience for your engagement.